Okay friends, now let us talk about the entry of a protein from the cytosolic environment into inside the mitochondria. So what we have this protein. This is a green colored protein along with this red which is a part of the protein, which is a signal sequence. And the protein comes in, it will attach with the receptor of a Tom complex. As we can discuss, that Tom complex is present in the outer membrane of mitochondria and attaches with this receptor. And you can see here this protein is not folded properly. Instead it will bind with the cytosolic HSP70, which is the chaperone proteins. The chaperone proteins are actually chaperonin proteins. Actually the chaperonin proteins are helping this unfolded proteins to be folded, to be misfolded, to protect it to be misfolded. Okay, so after that what happens, this Tom complex moves or migrate through this outer membrane, and it finally align its translocational channel with this translocation channel of TIM23 complex. As we know, this TIM23 complex is present there because it has the extension arm which is embedded into the outer membrane, and then the translocation channel alignment is done. Then the protein signal sequence attaches with these TIM23 complexes, and you can see in this situation there is a voltage gradient or the chemical gradient, electrochemical gradient, which is going on in the periplasmic in the intermembrane space along with the inside the mitochondria. Okay, a membrane potential is generated. And why this membrane potential is important? Because this membrane potential is actually helping these protein molecules to pull inside the mitochondrial matrix. Okay, so what happens in this first step, when the signal sequence attaches with the TIM23 complex, here we need an ATP molecule or energy source. So we have the ATP molecule. ATP is hydrolyzed into ADP and inorganic phosphate. As soon as it is hydrolyzed, the chaperone proteins or chaperonin proteins are released, and the protein molecules is entered into this periplasmic space. And whenever it is entered into the periplasmic space, it is attached with the TIM23 complex via the signal sequence, as we can see here. As soon as it attaches, then the membrane potential is generated, and as soon as the membrane potential is generated, it is translocated through this through the translocational channel which is present in TIM23 complex. It is slightly translocated towards a matrix, and the signal sequence travels fast because it is the guidance, it is the engine part of the protein which drives the protein inside the matrix. So the signal sequence comes fast, and as soon as the signal sequence comes in, there are different protease enzymes which are present inside the mitochondrial matrix, which is going to cleave the signal sequence because they do not need it anymore, because signal sequence done its job very good or properly. And after that cleaving of the signal sequence, there are mitochondrial HSP70, and these chaperone proteins again bind with this protein. And actually these proteins, as we have talked before, this protein present in this TIM23 complex, and it is actually helping the protein to be pulled inside the mitochondrial matrix. And again they need energy to do this pulling work, and they take the energy from the ATP hydrolysis. They again hydrolyze the ATP into ADP and inorganic phosphate, and via using the energy of this ATP hydrolysis, they pull this protein throughout the TIM23 complex inside the mitochondrial matrix. So that's how the process is done, and the protein is pulled inside the matrix, and again the Tom complex and TIM23 complex is dissociated, and they are ready for several round of protein translocation. So that's how the whole process is going on. So again I'm repeating it. So protein comes in, signal it will attach with the receptor via the signal sequence. Receptor recognizes the signal sequence. If the signal sequence is the proper one, then the further steps is going on, and if it is a wrong sequence, then this protein is denatured or cleaved. Okay, the protein is released actually, and it will be ubiquitinated, and finally it will be degraded via the proteasome system. Now if it is the right signal sequence, then the signal sequence is being transferred from the Tom complex into this TIM23 complex after the alignment of the channel, translocation channel. And then the ATP hydrolysis work occurs, and they take the energy for doing this, and the protein molecule is dragged into the periplasmic space. After the dragging it into the periplasmic space, there is a membrane potential which is generated here. After the membrane potential generation, the simple part of the first part of the signal sequence transported inside the matrix. As soon as it is transported, it is cleaved by the protease enzyme which is present inside the mitochondrial matrix. Then it goes through another round of ATP hydrolysis, because they need energy to pull this other part of the protein inside the matrix, and the HSB70 protein of mitochondria is doing the job by hydrolyzing, by taking the energy after the hydrolysis of ATP, and they finally pull that protein inside the mitochondrial matrix, and then the protein complex of Tom and team is dissociated. Okay, so that's the overall process of protein translocation from the cytosol into the mitochondria.